we've begun to find our feet in the very colourful world of representations, and maps were useful for us for distinguishing different ways in which something can be about something that's not present. And now we're going to dip in and go straight to an area in which representations feature very strongly, and they contribute to our understanding of the brain, which is always good. But I want to focus on three particular instances, which are all very similar in character, where we can look at the arrangement of the brain, specific parts of the brain, and say that that is a representation of something. But it's going to be a representation in a very specific, limited sense. So we're going to start with the connection between the eyeball and the back of the brain, the occipital lobe, which we've met before. As you know, the retina is a skin-like surface on the back of the eyeball, and it's directly connected to the brain, and we can follow those connections. We saw that the plumbing is kind of weird, and I'm going to simplify and just ignore that for now. But we can follow the direct traces from the back of the eyeball on the retina to primary visual cortex in the occipital lobe at the back of the brain. Now, when we do so, what we find is that two locations which are close together on the back of the retina project to two places in primary visual cortex that are also close together. So there's a preservation of the kind of spatial layout that arises on the retina. It's maintained to some extent in visual cortex. And if you recall what we met in the last video, we distinguished between symbols like the airplane or the prohibition sign, and more complicated cases like the distance on a map, representing distance on the ground. And we also met the imprint, where we had lipstick traces on a glass, or the imprint of a hand on a wet mold of clay. Now, think about this in those terms, and I think you might agree that this mapping from the retina to the primary visual cortex is more like an imprint, in that it has the same spatial characteristics with some alteration. And there's a direct causal link we can follow, we can trace from one to the other. We don't have to abstract. So in some sense, what's presented on the retina is represented in visual cortex. So that's one sense we might use that. Let's have just a look again at the mapping in more detail here from the retina to the back of the brain. Here I'm showing you a schematized vision on the top of the retina, and there's three areas outlined there. The dark center in the middle is the fovea, and then there's a gray area around it called the perifoveal region, and then there's a much bigger white disc around, which is the periphery. This example is taken from monkey brains. On the bottom, you see a view of the brain, and we need to understand where this comes from. The brain has two hemispheres like this, and what we've done here is we've opened them up so that you can see inside of primary visual cortex. So the brain is closed, but we've opened it up, so the two surfaces you're seeing actually lie on top of one another. And there you can see I've colored it, or the maker of the diagram has colored in the areas in primary visual cortex corresponding to the three areas on the retina. There's a dark area for the fovea, a gray area for the perifoveal region, and a white area for the periphery. Now notice what's happened though. This is not a straightforward mapping. It's not like a mirror, like an image in a mirror. If we look at primary visual cortex, fully half of the area of primary visual cortex connects to that very much smaller area of the fovea on the retina. Meanwhile, the periphery, which is much larger in the retina, is shrunk down to be a very small part of primary visual cortex. You remember the imprint we saw in the last video of the hand in a wet slab of clay. We said that when you press your hand in, it leaves, as it were, an imprint of your hand. But we can stretch that piece of clay, and the hand is still perceivable. We can still see it's a hand even as we make the fingers longer or shorter, or the hand base narrower or wider. So this kind of distortion, this stretching and compression has gone on. And it kind of makes sense, because what this means is that the part of the retina where most receptors are, where our sight is most acute, the fovea, has more brain real estate with it than those parts where we're gathering less information about the world. So that's vision. Now if we turn to the sense of touch, and we're going to treat touch as if it were a simple sense here now, we're only talking about light touch felt on the skin. We met some complexities last time. Here we're looking at a cross-section through the brain in the primary somatosensory cortex. Up in the top right of the screen, that's that blue fold on the brain. It's at the very front of the parietal cortex, and just beyond that is the boundary between the parietal cortex and the frontal cortex. Here again, we can follow the pathways directly using neuroanatomical methods. We can follow pathways from the skin, where the receptors are, all the way to a corresponding location in primary somatosensory cortex, which means that the surface area of the skin maps to this particular fold in the brain. Now here, the mapping is a bit more complicated than from the retina, because the retina is just basically a disc, whereas the three-dimensional outline of the skin is kind of hard to lay out in map-like format. So there's a few cuts and breaks here in primary somatosensory cortex, but you can see there on the diagram that different parts of the primary somatosensory cortex correspond to different parts of the skin, and that there's been some significant changes in size. That is, some bits have been compressed and are smaller, and some bits have been enlarged and are larger. So you can clearly see that the lips, particularly, and the hand would appear to be very, very sensitive, while the elbow and the heel just don't get a look in. You're not very sensitive on your elbow and your heel. So we get something like a slightly disjointed map of the body on the primary somatosensory cortex, which we can understand because of its direct connection to the surface of the skin. Some people like to talk about this as a representation in the brain of the surface of the body. But the term representation here is being used again like the imprints that we saw, not like the symbols or like the distance on the ground. We can actually look at these compressions and distortions to see what's happened as we move from the skin to the primary somatosensory cortex, and that gives rise to this very famous illustration. This little homunculus has been assembled to show the relative changes in size of the areas. Again, you can see the mouth area, the tongue and the lips and the hands are very, very sensitive, whereas the elbows, knees and heels just hardly feature at all. So we've seen this mapping for vision now from the retina and for touch. The third sense where we see this is hearing or audition. And this shows you a cat brain, and the grey area on the left is the primary auditory area in temporal cortex on the side of the brain. And the rather complicated set of dots there shows you that the neuroanatomists have gone in and they've registered the sensitivity of individual nerve cells here to frequencies in the sound that the animal is hearing. And you may remember when we looked at the ear, the receptor surface in the ear is the basilar membrane, in which low frequency sounds are registered at one point and high frequency sounds at another. We find the same mapping, that is a range from low frequency sensitivity to high frequency sensitivity, in the temporal cortex of the animal. This is a cat, as I said. So this is a tonotopic map, mapping from the sounds, the frequency content of the sounds, which are distinguished on the primary receptor surface in the ear, and which emerge again in primary auditory cortex. There will be again some distortions. So we've seen it in these three sensory modalities. We find nothing like this in taste or smell. These are all the three principal primary sensory cortices. So we've got the somatotopic map of the surface of the body, the retinotopic map from the retina to primary visual cortex, and the tonotopic map from the basilar membrane in the ear to primary auditory cortex. In each case, there's something that these all have in common, and it's very important that we understand that. The sensation is going to be occasioned by the play of energy on the retina or in the ear or on the skin. And that play of energy has a certain structure which is spatially distributed, spread out. And that spreading out or distribution of the energy is somewhat preserved as we look at the sensitivities of the primary cortex. There are distortions because some aspects of a signal are more important than others. We saw this with the lips and the heels, or with the fovea and peripheral vision. So there's a basic principle here, which is well known, has been well studied, and yet there's lots of occasion to introduce misunderstandings here. Because of the diagrams I showed you, to some people these clearly show that there are representations of the body in the brain. Now that word is going to become contentious. There will be other claims about representation made, which you might like to consider with a certain degree of caution, or you might like to consider as hypotheses. But those claims will be made on a completely different basis from these. These, as I said, are much more like the imprint. So there is a correspondence of some kind, of a particular kind, between the activity on the primary sensory cortex and the activity on the receptor surface. What these do not have is that notion of an ecosystem of signs or a context which gives them meaning. You remember that signs like an exit sign or a prohibition sign or even a letter like pi only mean something because they function within an ecosystem of related signs. This is not obviously the case for these particular things. So when we're claiming these, if we use the word representation for these mappings, we're using it in this limited sense of the imprint. But we're very confident, which is unusual in cognitive science, because we've traced the anatomical connections and we can say what we mean by representation. So we've got a translation here, a causal link from the receptor surface to the brain. And that's a start. Peering into the brain is going to be harder, but this is a useful sense of representation and one that can be distinguished from other more hypothetical and fanciful senses.